This video is a showcase of the Biotech Scholars Program, which allow young high school students taking the Introduction to Biotechnology course at Mass Bay Community College. Framingham High School students share their hands-on experience in a biotech lab during the spring of 2023 semester. Come and see what we have learned as young scientists on the cutting edge of innovative technology. These are examples of stuff you should never wear in the lab. You should always have closed-toe shoes, and you should always have clothes that cover your whole skin so there's no exposure.
Okay guys, let's walk over to the biosafety hood. So, we use the biosafety hood at this site to prevent contamination when setting up for experiments and pathogen cells and such. And we have to use an aseptic technique to make sure there's no contamination also. And we set up most of our experiments here. Here are the incubators we use to keep the cells we experiment on at an ideal temperature as needed for whatever experiment you might do. Usually they are set at 37 Celsius, which is the internal temperature for our body.
What is being done here is micropipetting. Micropipetting is what we use to transfer our cells, buffers, and even serums to different test tubes. On the micropipettes, there are dials you can move so the correct amount of the sample is taken, not too much and not too little. We transfer them into a separate tube, close it, and then put it into the centrifuge. We add another sample with the same volume to balance it in the centrifuge. This is because, for safety reasons, we need to balance the sample in the machine.
The nail drop is an extremely vital tool in our process of detecting the housekeeping genes and quantifying the amount of RNA we had isolated from our samples. The nail drop is an instrument that offers the ability to quantify or find the concentration of a specific molecule, such as double or single-stranded DNA, RNA, proteins, etc., in a given substance. For our experiment, we were looking to find the amount of RNA that was in our samples. The nail drop results had informed us of sufficient amounts of RNA.
PCR, or polymerase chain reaction, is the process used to amplify DNA. Then amplifying that to be able to use the DNA in multiple experiments when you originally had small samples. PCR is split into 3 steps: denaturation, annealing, and extension. First is the denaturation of the template into single strands. Second is the annealing of primers to each original strands for new strand synthesis. And third is extensions of the new DNA strands from the primer.
We want to acknowledge and thank Dr. Singh, the instructor of Biotech 101, for being such a hard worker and being an awesome professor. We also want to thank Colleen, Linda, Frank, our great TA, Mass Bay, Framingham State, and Dr. Dinklacker.